All right, so the paper we're going to do today is this one, Prediction of Future Input Explains Lateral Connectivity in the Primary Visual Cortex by Klavinskis-Whiting. I probably butchered that name, sorry. So one thing to note here right straight off is it is very unusual that we're doing this type of a paper because this is a purely computational modeling paper, and we're usually doing primary research papers. So you can say, well, what the heck is with that? And the reason why I thought it would be good to do this paper is because this paper is actually it's collecting the results of a whole bunch of primary research papers, and it is trying to explain the connectivity versus function results of those primary research papers. So I'm going to take the first part of my this talk, and I'm going to go through 4 of these primary research papers that it references, because it's going to present a model. This paper today is going to present a model that that they claim will explain the synaptic connectivity that is being seen by these primary research papers. The other way you can think about this is, from an aspirational neuroscience point of view. Is a visual cortex paper, and I want to argue during this that the visual cortex and its learned behaviors, its learned ability to process the visual scene, is absolutely learning. It is. I would say that there isn't a hard distinction between learning and memory, and all of the synapses that we're going to be talking about today are, at least if this paper is correct, are the result of learning based upon visual input. So if this could be decoded in a major way, if the synaptic connectivity could be decoded in a way that would show that it's predictive of the functioning of the visual cortex in a non-trivial manner, then that would also count as winning the aspirational neuroscience memory decoding challenge, at least in my mind, and we can debate that later. Okay, so this paper references heavily these 5 primary research papers, and a lot of them might seem familiar, because we've actually 3 of them we've actually done journal clubs on before. So that's the other reason why I think this paper is great. One is Iacaruso 2017. There's a Kossel 2015, Ko, Ding, and Ron. I'm going to go through those in order of dates now. Okay, so first primary research paper is this one from Ko in 2011, and I want to make clear what they did from a technique point of view, because I think it's very important to understand. What they did is they did calcium imaging in the primary visual cortex of a mouse, layer 2-3, so that they could build up the receptive field properties of the set of cells in that imaged region. And then they sliced that part of the visual cortex out and did in vitro patch clamp, as shown here. Hopefully as shown here. So they were patch clamping these 4 cells that had previously been imaged in vivo. And by doing that, they can inject current in one of the cells and see if the other cells respond to that injected current, which means that there would be a synapse between the two or multiple synapses, and they can measure the strength of that synaptic connection. And so what this paper was doing was it was saying, what is the relationship between the receptive field properties and the connections between these neurons? And they got these major results here. They were showing the mouse full field gratings, drifting gratings in different directions, and they would look at pairs of neurons, and they would say, if this pair of neurons if the orientation of the bar that it is sensitive to, the orientation of the grating that it's sensitive to, of neuron A and neuron B, okay, if the difference in orientation selectivity of those two neurons is 0 degrees or within kind of have the same orientation, up to 22.5 degrees on either side, then there is a 0.35% probability that they are synaptically connected when they look in the in vivo patch clamp. And if the two cells, on the other hand, are offset by their tuning responses are offset by 45 degrees thereabouts, then there's a lower probability, about 0.25% probability, that there will be a connection. And if they're orthogonal, go ahead.
Well, Randall has a hand raised, and I also you're saying you're speaking in very tiny fractions, 0.35%, but I think you meant proportion, like a third and a quarter, right?
That's right. Okay. I'm not saying 35.
And Randall?
Thank you.
Yeah, it's not a big one. I was just curious, in their paper, were they able to tell during their patch clamping for sure that these were direct synaptic connections, or could this have been through like two synapses or something? Was that obvious from the patch clamping?
I don't think you'll ever see a multiple synapse, because driving one would never cause action potentials downstream. So you're only going to see through a direct synaptic connection with the patch clamp.
Okay, thank you.
Okay, so thanks. This is so there is a particular pattern that is showing up here, and this is the pattern that they're going to look for in their computational model later on. They also did this for direction selective neurons. So direction selective neurons means that the grating has to go in one direction and the cell responds, but it goes in the opposite direction and it doesn't respond. That's a direction selective cell. And in this case, 6 out of 13 neurons with the same direction selectivity were connected. So almost half of the cells with the same direction selectivity were connected. And it's less for if they're offset by 45 degrees, less if they're offset by 90 degrees. It actually goes up a little bit if they're going in opposite directions. And so this is it's a signature of the connectivity in related to the functional properties of these cells that was found in this paper. The other thing they found in this paper is they showed the mouse natural stimuli movies that were moving around, and they just plotted the overall correlation of firing. So if two cells were firing together with a correlation of 0.3, then there was a 50% probability that they were connected to each other. But if there was no correlation, there was only a 5 out of 37 neurons that were connected to each other. Okay, so that's the Ko et al. Paper. there's this 2015 paper from Caussel. They use the same technique here, and but in this case they go further. They instead of just using full field gratings to measure the whether a cell is orientation selective and to what angle and direction selective, they use white noise stimuli to map out the receptive field properties. Where it activates in red is where if a white dot were to blink in front of the mouse at this spatial location, then the neuron would be more likely to fire. If it if a white dot were to blink in this blue location, that neuron would be less likely to fire. So the red is the excitatory receptive field and the blue is the inhibitory receptive field. So this is giving an example of two neurons that have a very similar overlapping receptive fields in space, as well as the same orientation tuning. And this is an example of two that are almost anti-correlated. They're actually looking at the same space, but the red is where the blue is in one, and the blue is where the red is in the other. So how does this correlate with connection probability, whether they have a synapse between them or not? If the spatial receptive fields are correlated a lot at the 0.8, 0.7 level, then the connection probability is actually quite a bit, 4 out of 6 neurons. And it has this, the more correlated the receptive fields are, the more likely there is a connection. And they were also looking at the overall strength of the connection. And that strength of the connection goes up the more the neurons are correlated. Okay.
Ken, can I ask a quick terminology question?
Yes.
So these cells that you're showing right now, like direction selectivity or orientation selectivity, that kind of cell, with these typical, say, for example, U-shaped responses and whatnot, this is all still, these are all considered so-called simple cells, right? Not the complex cells, or is it the same for complex feature cells in V1?
That is a great question. These are all layer 2-3 cells. So the current paper does talk a bit about complex cells versus simple cells, because they measure that. These 4 primary research papers, I don't think they measured complex versus simple. I know it sounds like something that they should have, and most of these are kind of assuming that this is a simple cell receptive field. So I hear what you're saying, Randall. So if I look at this, these receptive fields essentially have to be considered as simple cells. Yes. Okay. It's a good point, but I don't have a good answer to that. In the sense that they do one measurement of modulation to see if there's so just bring everybody up to speed. The difference between a simple cell and a complex cell is complex cell in Hubel and Wiesel terms are ones that are not very sensitive to the spatial location of a bar. It'll activate to a bar over a larger region. And in this case, they're not making that distinction in these papers, I don't believe. Another paper, primary research paper, was this Iacaruso et al. 2017. Now they used a different technique, a more powerful technique, 2017, more powerful technique, which was to look at the calcium dynamics of individual dendritic spines. And the individual dendritic spines calcium boosts up whenever the presynaptic cell is firing. So by looking at a whole bunch of individual presynaptic spines, or the inputs, they can build up the receptive field properties of a lot of the inputs onto this cell. And then they can look at a little piece of the dendrite of the actual cell and measure its calcium response and get a pretty good idea of what the whole cell is responding to. And so using that, they were able to build up receptive fields of not only the dendrite, so this dendrite, it can be considered the target cell, the postsynaptic cell, the main cell that they're looking at. If its receptive field is oriented up and down, shown here, then they can ask, does the spine's receptive fields, the inputs to this cell, are they structured in such a way that are they structured in a particular pattern? What they find is that if the spine receptive field, the input, is in the same, sensitive to the same orientation, but is offset in the direction of that orientation from the main cell, then there's a large likelihood, 30% likelihood that there will be a connection between those two. There's all of these are spines, so they're by definition connections, but most of the spines are, so 48 spines were found to have the same orientation to within, the delta around 0 degrees plus minus 22.5. There was only 33 spines that were offset in this way, but at a 45-degree angle. And then there were 16 that were offset at an orthogonal direction. And so you get this pattern of the connectivity. If you look at spines whose receptive fields are offset not along the line of the main cell's orientation preference, then if there is a connection, there's less connections, first of all, and those connections are not weighted by the orientation of those inputs relative to the target cell. Okay, so that's a signature of V1 connectivity that this paper is going to try to explain. And then finally, there's this 2020 paper by Rossi et al. That uses yet again a totally different technique, even more powerful technique than the 2017 one, that uses a rabies virus injection on a single target cell in layer 2-3, and that retrograde labels all of the cells that are connected to this cell, coming from layer 2-3 and actually from layer 4. So it's whereas these other papers were getting a tiny subset of the inputs to a target cell, this is getting almost all of the inputs, at least in the cortex, to this target cell, or a good fraction, I should say. And by doing that, they were able to figure out not only the excitatory but the inhibitory cells that are connecting to this, and to plot the offset in receptive fields between the excitatory cells that are projecting to this target cell, and the inhibitory cells that are projecting to this target cell. And when they look at a direction selective cell, so this is in this case, this is a strong direction selective cell, DS, direction selective, whose orientation is looking for a bar that is moving to the right. The excitatory inputs are offset in space ahead of that bar, and the inhibitory are offset. It's what we mean by ahead, but the bar that's moving in the preferred direction is going to hit the excitatory ones first, and then the inhibitory ones. And this is an explanation of why it's sensitive to a bar moving in one direction, but not sensitive to it moving in the exact opposite direction. So this is a signature of connectivity that is also known to exist in primary visual cortex. Okay. Okay. So any questions about those? So this paper that we're doing is try to explain all of these. Yes.
Yeah, I just want to get a sense because it's clear what the excitatory inhibitory offset and delay is for. Is it also the fact that there are a similar number of excitatory and inhibitory cells involved, or is this a totally different number in that each population in V1?
So it's definitely not the case that there's a similar number of excitatory and inhibitory cells. In this paper, they argue that it's about 90% excitatory versus 10% inhibitory in layer 2-3. In terms of the synaptic connections here, I would suspect that there is a lower number of inhibitory connections, but I don't know.
From just looking at this as a model of detecting direction selectivity, or maybe that's the wrong way of looking at it.
No, I think it is the right way of looking
can
at
you
it.
think about why there's this difference in the number of excitatory and inhibitory cells? Is it because they're only needed for this direction selectivity, but not for detecting other things like orientation and whatnot? I'm just throwing something out there. I'm just wondering.
Yeah, that's a great question. I have never thought of that. It's but in my back of my mind, I'm like, the excitatory cells are the ones that are mainly passing positive information, and the inhibitory cells are. That's not a good explanation, though. I don't know.
Okay.
I don't know. Although yeah, and I have to say that this paper is not going to say anything in regards to that as well, because they baked it in. They baked in a lower number of inhibitory cells. Okay, so what does this paper do? So this paper tries to this paper is making the claim that let's wait a little bit. So here we show that the functional specificity of V1 connections emerges naturally in a recurrent neural network optimized to predict upcoming sensory inputs for natural visual stimuli. Okay, so they propose this temporal prediction model, and they show that it reproduces the complex relationships between the connectivity of V1 neurons and their orientation and direction preferences. The tendency of highly connected neurons to respond more similarly to natural movies, and the differences in the functional connectivity of excitatory and inhibitory V1 populations. All of those results that I just showed. So that's what they're going to try to show. Hold on. So here is the model. It's a very simple model that they're using. The model is a So this is the model. There is an image that is being put into the model. It's I think it's 36 by 36 pixels. Those are fully connected to a set of neurons here. This is the quote unquote hidden layer. Those are the neurons that are supposed to be the layer 2-3 neurons that we've just been talked about. So the responses that they're trying to show and the connectivity are among these hidden neurons here. They make reciprocal connections within this layer. And then, oops, and then there is a decoding network. Now, this decoding network is not supposed to be biologically relevant at all. Okay, this decoding network is just supposed to be there to say, to train this network of pseudo layer 2-3 visual cortex neurons to make sure that it has enough information to reliably predict the next frame in the movie. And in this case, that would be 40 milliseconds into the future. Yeah, yes, Randall?
Yeah, I'm sorry if I'm getting ahead of you, because maybe you're about to go there. But one of the things I was trying to grasp from reading the paper is how much I should read into the model that they built in terms of its biological plausibility, and how much we can say about how the same sort of thing can happen in V1. And so part of that was, okay, some basic things like this has a state vector, and I kind of interpret that as like a firing rate or something like that, right? So maybe it's not a spiking, it's not trying to be that precise.
Not a spiking. That's
and they're using backprop for the training, but they say that they're, they kind of alluded to, but there are ways that you could implement a similar treatment of the prediction error biologically, but then they don't get into the details. Maybe I should have checked the references about how that would occur, because you do, if you want to train the system to do prediction, you do have to somehow compare the actual outcome and the predicted outcome, as you show here with this decoder network, and make that have an effect on those weights, right? So I was trying to wonder or figure out how would this happen in the biological network, but it's okay if you can't get into that. That was something I was really
and I think
about.
It is a different paper, because what they're essentially showing is they're saying we can predict the receptive field properties of layer 2-3 visual neurons and their interconnections only by assuming that there is some mechanism
Right.
that we don't even know about.
No, I realized that. I totally realized that. Yeah.
And
this was more of a matter of curiosity on my end of because in the paper they also sometimes talk about how this could be something that is based on evolution versus on learning, and they don't quite stake their flag there. They say, this paper isn't going to make a firm claim about one or the other. And yeah, so I couldn't help wondering how it would work.
Yeah, and I wonder as well, and they do not in this paper, but they have other papers that they speculate about that. And it would be essentially be feedback connections that are not shown here. Okay, so we got these. It's important to understand that there's a set of weights here, W input. There's a set of recurrent weights, W. Hold on a second. Hide that. W recurrent, and there's some biases. these things are going to be, these weights are going to be, let's see. I'll get to that in a second. There is also these units here that are supposed to be the layer 2-3 units. 90% of them are set to be only excitatory. So if they ever go negative, they just get clipped at 0. And the remaining 10% are designated as inhibitory units. If they ever go positive, they get clipped at 0. So there's 90% excitatory in here and 10% inhibitory by design. And the output here, this V vector, that is going to be compared to the next frame, which is 40 milliseconds in the future of these natural images, natural movies of natural scenes that are kind of animal like panning over leaves and things like that. Okay. The loss function here is essentially saying I want the prediction to be as close as it can to the to what the actual image will be 40 milliseconds into the future, and it adjusts the input weights and the recurrent weights and the output weights to achieve that, and it also has a normalization function, which turns out to be an L1 regularization function, which turns out to be quite important, that limits the total sum of the weight values to be, tries to make it low as well. So the back propagation algorithm is adjusting the weights to make it so that that it is able to predict 40 milliseconds in the future what the image is going to be, but it also tries to limit the total sum of the weights that do that. Okay, so they run that training on all these natural images, and what do they get? Okay, so there's like 6 or 7 findings here that I'm going to go through. Most basic finding is that the model units, so these are in B here, these are the receptive fields of neurons that were learned in the model. And in C, this is the receptive fields that were measured for neurons in the mouse. There's some way that they equalize the size of these. Yes, Randall?
Yeah, just a tiny thing. I was just wondering why they went ahead and pre-processed those mouse responses with a Gaussian filter, if the result wouldn't have been the same if they just left it as was.
I suspect that what they typically do with my understanding is what they typically do to get these receptive field properties is they throw a whole bunch of white noise stimuli, and they add up responses for white noise stimuli. So you get a really noisy image, and if you don't blur it a little bit, then it looks weird. I suspect that that's why they did that.
All right.
Okay, so in general, it's giving the same types of responses as the real thing. The model also learns to have both orientation-tuned units and direction-tuned units, just like a visual cortex layer 2-3 does. Okay, so in the model, these are, so this first one is a orientation tuning. These are all measured by drifting gratings. So if the grating is drifting in the 0 degrees, or if it's drifting in the 180 degrees, it gives large responses in both directions. But if it's drifting in 90 degrees or 270, it doesn't. This is one neuron. This is a different neuron that is direction selective. So it's only if it was drifting in the, what would this be? 220 degree direction does this respond, and even if it's going opposite, it doesn't. you get the same types of receptive fields in mouse V1. The model shows a similar amount of simple and complex cell responses. So this is the one thing that I will talk about, simple and complex cells. And they measure simple versus complex by talking about the modulation ratio to a grading. You can imagine if you really have a complex cell that doesn't care where a bar is that's moving through its field of view, then the modulation as it moves through would be 0. So that would be kind of a pure complex cell. And if it really cares exactly where it is, it would be a pure simple cell. And just like in V1, so the black is model measuring all of their cells on this complex to simple axis. And this is this is a histogram. So it has a similar distribution of complex to simple as the real mouse V1 does, which is kind of cool. Okay. None of these we're talking about the connectivity. Now we're going to talk about connectivity. In order to talk about connectivity, we have to define what connectivity means in a network like this, as opposed to the measurements that were done in the primary research papers. In the primary research paper, there's either a synapse or not a synapse between the two cells. There's either there's either a synapse or not a synapse. In this case, units were defined, so these model cells were defined as connected if their connection strength exceeded the 95th percentile of connection weights in this recurrent weight matrix across all pairs of units. So that's how they define it. They say that due to the sparse nature of the recurrent weight connectivity matrix, this threshold is equated to rejecting the very low or 0 weight connections. By the way, why is the recurrent weight connectivity matrix sparse? Well, it's sparse because they had that L1 regularization that punished the network if it had too many weights. Okay, so so finding number 4, what they do is they look at the connection probability of the model neurons relative in black here, relative to mouse V1 actual connectivity. This was from the Ko et al. Paper that I showed before, and it's giving the same pattern of connection. They also look at that for orientation differences, and they look at this for direction selective cells, and they're seeing not only the same pattern, but they're seeing this kind of U-shaped curve as well, which is, I think, pretty cool. Number 5, finding number 5, the model shows the same relationship between response correlations and probability strength of connections. Remember, this was for mapping out the receptive fields and how they overlap with each other. This is saying, hey, if we have two cells that overlap closely with each other, then there's more of a probability of connection. Again, the black here is the model, and the gray is the actual V1 connectivity results from Hassel et al. Finding number 6, the Iacaruso 2017, they try to replicate that. They see that you get, if you look for longer distance connections, cells with the same orientation preference are preferentially connected, and it has the same pattern of connection. So if we look at the presynaptic fraction of inputs to a model cell. It's about 30% of those are aligned with the target cell's orientation preference, just like we see in mouse V1. And just like in mouse V1, if we're looking at not in the direction of the orientation direction of the target cell, it doesn't really care. And so what is this useful for? It's useful for enhancing responses to elongated edges. It's seeing the same thing in its model. And finally, oh, and this is the exact same thing that they were seeing from Iacaruso. And then they're looking at the excitatory versus inhibitory for direction selective cells versus non-direction selective cells. And just like in the real V1, they see an offset between the red, the excitatory inputs, and the blue inhibitory inputs in a way that explains the direction selectivity. By the way, they don't talk about a time delay in this. And so their model is a recurrent model. They could measure the time delay, but for some reason they don't measure that time delay. It's interesting that they don't.
Sorry, I thought they did mention it somewhere, but maybe I'm misunderstanding what they were showing. Because obviously they were comparing across 40 millisecond differences and doing prediction for the next frame. So I don't know, maybe I just read that as being implicitly the time delay, but that's not necessarily clear.
So let me say what I would have thought they would have had to do in order to do this. I would have thought they would have had to bake into the model a time delay from the inhibitory cells, which they could have done, but I don't believe they did. And so if I'm understanding correctly, the idea is that the model of how the V1 would be doing this would be to make sure that the inhibitory synapses have a delay associated with them. In any case, I don't think they talk about that, but I could be wrong. This is just showing that spatial layout. They also compared this to different models. I'll go through this quickly, running out of time. So they compare their temporal prediction model, which is this one right here, its ability to predict the next frame, how well it does in doing that versus other types of models. And so they used a sparse autoencoder and a denoising and an inpainting. Basically, inpainting means all of these were trying to predict the current frame, but in a way that got rid of noise, or in a way that got rid of blanked out regions, or in a way that used a small number of neural responses in the hidden layer. That's what an autoencoder does, a sparse autoencoder does. None of these were as good at predicting as the temporal prediction model. They compared it to a bunch of other ones. Those are probably not as relevant here. The more interesting thing is if we compare their model to the proportion of units that were orientation tuned or direction tuned or non-selective, mouse V1 is here, and their model, their temporal prediction model, is showing about the same proportions, whereas these other models that weren't doing temporal prediction, they were just trying to predict the current frame, show very, very different responses. They don't need direction selective neurons because they're not trying to predict into the future. They also look at, and this is probably the main that I'm interested in here, the correlation with the V1 connectivity stuff that we just showed. All of these different results of connectivity in the model versus connectivity in mouse V1, they compared this with these other models, and the temporal prediction model is much more correlated from a connectivity point of view than all of these other possible models of V1. They looked at connectivity scores versus how far into the future, and maybe this is what you were talking about, Randall, how far into the future they were requiring the model to predict. Did they require the model to predict only the current frame, or 25 milliseconds into the future, or 50 milliseconds, or 75 milliseconds into the future? And when they were predicting 33 milliseconds into the future, they got the highest correlation with with what you actually see in V1.
So that graph predicting into the future, does this also tell us basically, let's say if you want to have a system that is detecting movement in a certain direction, does this tell us the speed of motion that it is best at detecting?
In this case, it's trying to predict all of these movies. So if there wasn't a bias in these movies, if they had a range of predictions, then hopefully the model would have been forced to have different velocity sensitive neurons.
Yeah, that's what I would assume you would have overall with plenty of training. Yeah. Okay.
Because
I guess I can't read that out of this. Yeah. Yeah. No, you're right. You're right.
Yeah. Remember, it's predicting based on a purely linear model, purely linear decoder of this layer 2-3 neurons. They also vary this L1 regularization amount, and if they allow the model to have a whole bunch of weights, then they don't get receptive fields that look like V1, but they also don't get as good of next frame temporal prediction. And so they make the argument that they chose minus 0.6 as the regularization, not just because it gave them the most similar looking stuff, but because it actually did the best at temporal prediction. And therefore, you might expect that type of a regularization be happening in whatever the learning algorithm that does this in visual cortex as well. And this is my last slide here. They have a bioarchive paper that is relatively recent that extends this to multiple layers, but because we don't have connectivity results from multiple layers that we can trust, like these results that I just showed you, they're really just looking at at how similar the receptive field properties are, but they don't have a connectome to actually compare this to. And that is my last slide. So I am going to attempt to find you guys again. Sure. Any questions? Ariel?
Yeah. Well, so that's a cool demonstration that you can take basically untrained random neural weights, run a bunch of videos over them with some fairly simple assumptions, and then you pull out something that ends up looking a lot like V1. That's sort of an argument for, there's nothing particularly special about what the visual cortex does. Initially, it just like develops those weights based on learning from sensory stimuli. I think I have a good idea, but maybe just to clarify, how do you think this ties into the, aspirational neuroscience decoding? I presume the argument you would make is that, the model you get at the end, either this trained model or what is exists in actual biological V1 has like learnt the environment compared to random weights to begin with. And if you can read from a connectome what those, the tuning, the orientation of any particular neuron is, then you've decoded a memory in a sense, or do you think about it slightly differently?
That is exactly what I am proposing here. I think that hypothetically, if somebody were to produce a connectome with calcium imaging of primary visual cortex or primary visual cortex and a higher order visual cortex, and they said we are going to mask out, we're going to hide half of the calcium, half of the cell's calcium responses. And you, based upon the connectivity alone, tell us as accurately as you can what the remaining calcium responses are. And you get something that is kind of like, several thousand cells calcium responses. You get to you predict their receptive fields like really, really good. I would find it hard to say that that is not in the spirit of decoding a memory learned behavior from a static connectome.
You're essentially, yeah, it's just you've determined what those cells are looking for. In a sense, if you wanted to drag it out one step into the environment, you can be like, this brain has learned to look for these kinds of edges, orientations, movements, those sorts of things is essentially what you're decoding.
So jumping on that, also, I was thinking about basically the same question that Ariel was asking, which is, so this paper is really cool. It's making the hypothesis that these connections are generated by being optimized for prediction of future input. And that's great. But where does that study fit in with memory decoding? I can see where I love the idea that we can say, yes, all like learning and memory, so closely connected, basically the same thing. So learning to set up or having set up a set of connectivity that produces a specific functional output, like being able to detect direction, orientation, that is just as much a memory as any other memory. And that's something that should count towards the prize. And you can take a biological sample, you can look at what's in there, the connections that you find, that from that you can determine, what does this do? Okay, now I found an engram. This thing is basically something that determines direction or something like that. Great. So I'm not exactly sure where the paper fits in, though. That's the one thing I'm still missing, because I love all the rest of it. I just don't know the hypothesis that this was achieved because it's trying to do the prediction. You don't really need that if you're just trying to find connectivity and what it does, right?
I think that's true. I think the main reason why, now it's my psychology that you're asking about.
Okay.
Why did I think this paper was important to this? My feeling was that there is a bias out there that would suggest that that this is all hard coded, that this is a primary visual area, and that maybe this is just genetically encoded. And I think what I was really trying to get forward from this particular paper is that no, that we shouldn't be thinking of these primary visual areas. Maybe we should when it comes to like fruit fly or something, but a mammalian primary cortical area is going to be learned.
Then there's still a bit missing for me here, because you see, the paper doesn't claim that it has determined that this is not hard-coded.
It's true. It's true.
They just show you, yeah, this looks like the system is doing prediction. And it'd be nice if we could like fill that in with the missing gaps, bits and pieces that say, yeah, we can show that this is a learned thing. And therefore, if you pick up on this, you're not picking on something that was hard-coded. You're really picking up on a kind of memory.
Well, for me, part of what that could be is like counterfactual scenarios. So I'm reminded of like the 1980s cat studies where they did horrible things like only show them horizontal lines or only show them vertical lines, or like sew one of their eyes closed for a while, and you get these like different dendritic patterns and connectivity patterns in the primary visual cortex. I don't remember what the behavioral sequelae, if any, of these studies were, but I do wonder what would happen if you did this same sort of study and you showed it like only weird videos with like only particular orientations or particular motion. Would you be able to clearly see like a very different connectivity pattern? Maybe the authors think that's too obvious and that they definitely would, and so that's why they don't show that as like a control experiment. But for someone who's naive to this, like me, I would enjoy a control experiment like that.
Yeah, although what they're trying to do in this case is they're trying to use natural stimuli, which means that they can't control it. But yes, they could absolutely control it in a way, but they would get a very boring visual cortex to look at. So maybe this is. I remember, but maybe this is just a lie, that the cats would like run into, if they didn't have vertical lines, that the cat would run into a vertical edge.
I have to go check the. Yeah, I remember them doing it. I don't remember what the behavior was, so we can check it. Come back.
Yeah, there's also this kind of I don't know if this is in visual cortex. I don't think this is in visual cortex, but I know in retina there's retina waves, and so there's going to be internally generated stimuli that are also interacting with what we would normally call a learning algorithm. And the way I look at it is the spirit of this is, can you? It is less about is it memory, is it learning? It is more about what can you get from a static connectome with regard to function. So I would even be drug in the opposite direction of saying, even if we were talking about a fruit fly brain, and the fruit fly brain was completely genetically hard coded, so it could be reared in the dark, which probably can. I know that they've done those experiments, and still have these visual responses. If you can decode these visual responses like perfectly, which I think you can,
So
then that says something deep.
to be kind of a little bit of a devil's advocate, then this would sort of become the memory, or no, the microcircuit decoding prize, or the feature decoding prize, or something like that.
Structure to function decoding prize.
Yeah.
It's a genetic memory at that point.
Exactly, and literally, I think there's a lot of heavy lifting being done by the word non-trivial. So at some point we have to decide what non-trivial means, because man, people are getting really good. I mean, at decoding, let's say, the fruit fly visual system.
For this one in particular, picking exactly what would be non-trivial, that might be there might be some work to be done there, because picking up on just a few simple cells might not be enough. You might have to do something a bit more difficult, like move up a layer. Yeah, I don't know. I'm not sure. But I personally think that this should count, but that's because I think picking up on circuits is interesting. So it doesn't have to be a memory.
I also think that anything in cortex has an assumption built into it that it is getting to a fundamental cortical learning role. And so even if it's a relatively simple function, if it's in mammalian cortex, the general rules that we're saying, like I would be really surprised if you were to look in auditory cortex, like secondary auditory cortex, and had some complicated receptive field of a phoneme, right? that you couldn't build its receptive field response based upon the synapses that were coming to it and their responses. Yeah, it's a general thing. Yeah, I know we're over time, so sorry I took so long going through that.
That was good.
Was good. And next time, I guess, is this true, Ariel? I'm going to present next time on another vision cortex.
In 2 weeks, I think. Where is it? I think you're currently slated for the 14th of April. So yeah, 2 weeks from now.
Great.
Sounds great. Yeah, this was fantastic. Are there any final words or anything else anyone wants to throw in, contribute, ask before we go? Oh, looks like that's all done. Thank you, Ken.
Thanks,
Thank
Ken.
you.
Thanks, everyone, for coming.
Bye all.
Yeah, bye.